Assalamu alaikum everyone. Today's paper was done by Donnermeyer and his group. It was published last month in the IEJ, titled A Comparison of Endodontic Microbiomes Associated with Symptomatic and Asymptomatic Apical Periodontitis by Next Generation Sequencing. The aim of the study is to compare the endodontic microbiome of teeth with symptomatic and asymptomatic apical periodontitis and test whether alpha and beta diversity differ between groups. Prior studies suggested possible microbial differences but were limited by small sample sizes, heterogeneous methods, and older sequencing platforms, making conclusions about symptom microbiome relationships uncertain. This study used a large sample and contemporary Illumina-based sequencing to clarify whether specific taxa or overall community pattern related to symptoms. The study used a cross-sectional observational design including 60 teeth, 30 in each group, with apical periodontitis confirmed radiographically and by clinical testing. After rubber dam isolation, surface disinfection, and access cavity preparation, a glide path was created and microbial samples were collected from 120 root canals using sterile NITI files size 2006 with one canal per tooth, and the one with the greatest sequencing depth retained for analysis. Bacterial DNA was extracted and the V4 region of 16 srRNA gene was amplified and sequenced on Illumina MySeq platform. Sequences were processed to generate applicant sequence variants and taxonomic assignment using the expanded human oral microbiome database. Diversity and community composition were compared between symptomatic and asymptomatic groups using multiple indices and differential abundance methods with multiple testing correction and patient as a random effect where appropriate. Microbial richness and diversity did not differ between symptomatic and asymptomatic apical periodontitis at either genus or applicant sequence variant level. Beta diversity analysis also showed no significant differences in overall community composition between groups on genus or ASV level. Both conditions shared most detected genera with broadly similar dominant taxa. However, differential abundance analysis identified 15 genera with significantly different relative abundances. Among the common genera, Fusobacterium was enriched in symptomatic cases, while Actinomyces was enriched in asymptomatic cases. Overall findings suggested that symptom expression is linked more to the shift in relative community composition than to the presence of unique symptom-specific pathogen. The study has several important limitations that affect how broadly the findings can be applied. Its cross-sectional design captures microbiomes at a single time point, so it cannot show how microbial shift over time might drive progression from asymptomatic to symptomatic disease. Illumina MySeq short read 16S sequencing limits taxonomic resolution at genus level, making it difficult to reliably distinguish closely related species or strain level virulence differences. ASV level results are also sensitive to technical artifacts and must be interpreted cautiously. Although the sample size is larger than in many earlier studies, inter-individual variability as tooth type, periodontal or restorative status, systemic conditions, and medications was not deeply stratified and could confound microbiome symptom associations. The study excluded patients with recent antibiotics based on short recall window, and the true impact of systemic antibiotics on root canal biofilms remain uncertain. And finally, all participants were recruited from a single center in Germany, so geographic and population differences in endodontic microbiomes may limit generalizability. In conclusion, symptomatic and asymptomatic teeth host diverse but broadly similar root canal microbial communities at genus and ASV levels, with no clear differences in overall diversity or community structure. No single pathogen profile explains the presence of symptoms. Instead, shifts in relative abundance, notably increased Fusobacterium in symptomatic cases and more Actinomyces in asymptomatic cases, may modulate host immune response and symptom expression. Clinical symptoms of apical periodontitis likely reflect community level composition and host microbiome interactions rather than specific microorganisms alone, underscoring the need for larger, high resolution, and longitudinal studies. And thank you.